Hi, my name is Victor Juan de Souza Lima, and I am an undergraduate student from Federal University of Santa Catarina. Today I'm going to present a study that we've been developing at the Laboratory of Stem Cell and Tissue Regeneration in Florianópolis, Brazil. The title of this study is In Vitro Comparative Study of Adipose Tissue-Derived Mesenchymal Stromal Cells from Canine and Feline Sources at a Low Passage. Mesenchymal stromal cells, MSCs, are a group of multipotent adult stem cells that have been used for regenerative medicine research and veterinary medicine. Most this is because of the therapeutic potential of these cells, such as the ability to modulate the immune system and to differentiate into another cell types to regenerate and damaged tissue. Several clinical studies were published evaluating the potential of cell therapy with MSCs, but besides that, only a few compare MSCs from canine and feline sources. Also, the cells can be obtained from many anatomical regions, but MSCs from adipose tissue display some advantage in comparison to other sources, since the adipose tissue from the abdominal region is easy to extract and there are some cell characteristics that favor cell culture. Said that, the aim of this study was to compare the characteristics of adipose tissue-derived MSCs from canine and feline sources at passage 1. So, to the isolation of MSCs, the adipose tissue was obtained from abdominal region of dogs and cats that went through elective surgeries such as castration surgery. MSCs were then isolated through mechanical enzymatic dissociation of adipose tissue, and after that, cells were cultured under standardized conditions. And of course, our study and our procedures that we did were in accordance with the Animal Ethics Committee from our university. We performed analysis of flow cytometry, scratch assay, measurement of proliferation rate, and cell doubling time. The flow cytometry results showed that MSCs from both sources display a particular immunophenotypic profile, being positive to mesenchymal surface markers and negative to hematopoietic markers. The proliferation assay was done along 120 hours, and we observed an increase in the number of cells over time, with no statistically significant difference between canine and feline MSCs proliferation during this time. The cell doubling time analysis showed that the average doubling time from canine MSCs was about 30 hours, and from feline was 37 hours. Despite that, no statistical difference was observed. In vitro wound healing was evaluated by cell scratch assay in confluent MSC monolayers. Wounds were photographed immediately after lesion and after 24 and 48 hours of healing and measured using ImageJ software. MSCs from both sources were similar at in vitro wound closure capacity. So these results show us that MSCs from canine and feline sources behave similarly at cell culture and have many mesenchymal stromal cells characteristics in common, such as expression of cell surface markers, cell doubling time, proliferation rate, and in vitro wound closure capacity. Also, the observed characteristics may indicate that MSCs from both sources have a similar potential and viability to be used in cell-based therapies, considering the process of obtaining and cell culture. But still, a higher number of MSCs donors are needed so we can better understand our initial results. It is with great pleasure that I acknowledge the Federal University of Santa Catarina, UFSC, for financial and institutional support. I also thank all my lab partners from research, our laboratory, for their contributions, especially the authors of this study. And at last but not least, thanks to CAPS and CNPq for supporting our group as well.